transcriptomics. That is a novel technology. And today's paper is published on 2021 on Nature Method. The title is SpaGCN, which is integrating gene expression, spatial location, and histology to identify spatial domains and spatially variable genes by graph combinational network. It's a valuable tool that could integrate the gene expression and the spatial location. And the spatial resolved transcriptomic SRT technology, which is a new technology, is enabled to comprehensively characterize the gene expression on the histology map, so we can know the gene expression pattern in the microenvironment. In this figure shows that this is a slice of mouse brain, and you can see the gene expression profiling, which was divided into several domains. This domain can show the different gene expression, and through the histology gene expression, we also can find the specific cells which is located in which area. Then in 2020, Professor Jacqueline Lodberg, who finds this important technology, he shown that the importance of spatial content to infer deeper biological meaning is unquestionable. Means that when we understand the gene expression on the histology map, we can understand the spatial gene expression. Then this is the history that we know until now, that how can we record the location and species of messenger RNA transcripts. There are two ways to understand the location and messenger RNA transcript. First is the imaging method. The second is sequencing method. The first one is that through the in situ hybridization, we can see the target messenger RNA will hybridize with specific probe. Then this probe will label with fluorophore labeling. Then we can see which specific messenger RNA's location. Then we will repeat several times to generate the gene-specific fluorophore barcode. Then we can understand which gene is located on specific tissue region. The second imaging method is that in situ sequencing. It's using the rolling circle amplification of target transcript. We can see that in this figure, it shows in three different kinds of color. Then after hybridization with short label probe to determine the one or two nucleotide of transcript sequencing. Then after imaging fluorophore of each location, we also can repeat this method several times to determine the messenger RNA sequencing. Through this method, we also can understand which specific messenger RNA transcript on specific location. And the second way to detect the location and the messenger RNA transcript is using the sequencing method, which contains the array method and the microdissection dissection method. Array method is that you use the array of spatial barcode probe, then image the barcode's location by in situ hybridization sequencing. Then we can see overlapped samples on array ligate messenger RNA to the probe, then using the NGS to capture the probe. The second one is to use the microdissection assay. We can hybridize the probe to the target messenger RNA, then imaging the specific region of what is interesting in this tissue location. Then we can release the probe from ROI with UV and capture via capillary. Then we can do the NGS sequencing of the capture probe. Through this method, we can understand the location and the specific messenger RNA expression. Now we have a new technology that's called the spatial transcriptomics. Before, we use RNA sequencing using the bulk RNA sequencing. We just dissect one specific tissue, then we perform the bulk RNA sequencing, which contains the total RNA sequencing in one tissue. And recently, we find that there is a single-cell RNA sequencing. We can understand what is the gene expression profiling in one single cell. So we can understand in this tissue it may be divided into 4 parts: the yellow part, green part, red part, and blue part. Then we can understand what is gene expression profiling in this specific tissue. And the other new technology is called the spatial RNA sequencing. It contains the histology and the gene expression profiling. Therefore, we can understand the red gene expression is located on which part of the tissue. So we can more understand what is the gene regulation in different parts of the tissue. Here you can see that the significance of spatial biology. In the left part is the histology analysis. We can see this one is human breast ductal adenocarcinoma. This is a tumor tissue, and from this slide biopsy of the breast cancer patient, we just see there are several tissues in this biopsy. What is lying beneath the surface? We don't understand what is the gene expression profiling in one single biopsy. The spatial biology helps us to understand, for example, in the red part, it contains a different gene expression compared to the yellow part. So we can understand each area of histology analysis. We can understand each area's the gene expression profile of each area. And the spatial gene expression has several advantages because it is a whole transcriptome analysis with the morphological content. So it will gain a comprehensive view of the disease complexity, and it will define the new biomarkers of disease, and we can understand the novel cell types or states. Moreover, it will map the spatial organization of cell atlas and identify spatial temporal gene expression pattern. For example, we can see this mouse brain shows in several different domains, and each domain will express several different genes. So the spatial data is mainly comprised of the two signals. It contains the histology image and the gene expression profiling. Then we can combine these two data together to get the histology and gene expression profiling. That's called the spatial transcriptomics. How can we perform the spatial gene expression? First, we have to prepare the sample, and using the imaging system, we can understand stand for HE stand for the morphology content, which will stand for the cell cytosol and the nucleus. Then we can know what kind of tissue we obtained. And after IF for protein code detection, we can, using the library construction, and we using barcode and library construction. After sequencing, we can see which gene is located on the specific tissue area. So this spatial capture technology will capture whole the transcriptomics analysis. It uses a special gene expression slide. You can see that this slide is very small, and we have to put the specific tissue on this small slide. And this slide will coated with capture with the capture area with 5,000 barcode spot. And each barcode spot contains a gene-specific barcode. It contains the special barcode and connected with unique molecular identifier, so we can know in this region what kind of gene is expressed. These specialomics were showing in this figure to let you understand how can we perform this experiment. First, we have to do the tissue section, then perform the histology analysis, then we HE stand to understand the morphology of each tissue. Then this tissue will apply to the oligoarrayed microarray slide. And this slide to back up of the dense compacted oligo-coated beads. These beads were further for important binding. Then the messenger RNA will diffuse to the slide surface and hybridize to the oligo-DT cDNA synthesis primer that encoded to the unique molecular identifier and the special barcode. Further, after reverse transcription, the cDNA will synthesis, and we can know which cDNA is produced and construct the library, then perform the sequencing, then we can know what is the special genes expressed in specific tissue area. So after computational analysis of specialomic data, which will map the sequencing end and to let us know to which gene expression after differential gene expression. So we can know in this area which gene is already expressed. In this paper, there are several studies already shows how to link the spatial domain. It means the histology area and how can we know the biological function of this specific area. There are several tools has been established. They first to use the gene-enriched gene expression profiling. Then we can understand these areas will call it as domains. And in 2018 and 2020, spatial DE and SPARC. SPARC means the spatial pattern recognition by a kernel. These two methods help us to define the spatially variable genes. And we can understand in this area which one is a spatially variable gene. And we can understand in this area the most important genes is expressed. Then it will examine each gene independently and return a p-value to represent the spatial variable genes expression. So in this paper, they aim to understand how can we define the spatial domain. Because before the method do not guarantee the spatial expression pattern, so it's hard to understand these genes for further biological investigation. So this paper, they also want to develop a new technology called the Spatial Graph Combinational Network, SpaGCN. And this network is a graphic combinational network approach, which will investigate the gene expression and spatial location and histology, and integrate these three these three data into the spatial results transcriptome and data analysis. Then, using the aggregation of gene expression of each spot from the neighboring spot, it will define the specific domain and coherent to the gene expression and histology. So here is the workflow of SpaGCN. You can see that from the data, from histology imaging and the spatial transcriptomics, it means the gene expression on spatial region. Then combine these two data and the XYZ axis. Then you can count the different region to build a graph that represents the relationship of all spots, which considering the both spatial location and histology information. Then after ReLU, this tool is a nonlinear activation function. It will then use the unsupervised irritated clustering. You can see the spatial data. Then, using the specific domain or non-specific domain, we can see the gene expression profiling that shows the specific gene marker, or we can define it through the meta-gene. Meta-genes means that there are several genes positive and several genes negative that will compose the different region of this histology data. So they first to establish the SpaGCN and evaluation. Then they want to further demonstrate the importance of the incorporating histology information. So in this paper, they use the open data set, which is from the human primary pancreatic cancer data set. It is generated using the spatial transcriptomic technology. So they have to compare the tissue structure and the spatial auto-regulations of gene expression. For the tissue structure, they use three tools. First is the relevance, which shows the cancer region, and ST-Learn and Bayes space. These two tools was used to understand the spatial clustering. So they will compare their data through these three tools. Moreover, they also want to understand what is the spatial autoregulations of gene expression. So they will compare the Moran I and Garris C statistics. So first, they use the histology. Then we can see the histology region in the red area shows the cancer cells. This one is the cancer cell. It contains stronger blue color because they have the enlarged nucleus. And the yellow part is the ductal epithelium, this one. And the green part is the intestinal. It shows in this histological image. Then after the relevance tool, it shows the cancer region. And you can see there are some overlap in this green color. The green dot is shows in the domain 2. And the domain 1 is the pink dot. And the domain 0 is the light green dot. Then you can see comparing the spatial cluster and the cancer region, you can see there are some difference between these three figures. And the cancer region is shows in the dark gray color. So they compare to this method and find that the spatial GCN is most similar to the histology region. So they find that this tool is very similar to the histology cancer region. Further, they want to observe what is the gene expression profiling of the spatial expression. Then they observe the KRT17, which is a tumor promoter, and the MMP11. It's a prognostic biomarker. And we can see that this gene expression was shown in different expression level. The red color shows the higher expression level, and the green color shows the low expression. Combine these genes, they find that in the KRT17 plus MMP11 with the negative expression of SERPINA1 shows the most abundant cancer regions in spatial GCN network. And this data is much like the cancer region analysis. So they use the different gene expression pattern that can analyze which location is the cancer area. Further, they use the spatial autocorrelation of the gene expression and find that Moran and Gerst statistics compare these two available tools, SPARC and spatial DE, in blue and yellow color, and find that in the red column shows the higher value of these two statistics, which shows that spatial GCN has higher auto-regulation of gene expression. So they have applied this tool to the human primary pancreatic cancer spatial transcriptomics data. They want to further observe what about this tool can be used in human most complex brain tissue. So they analyzed the LIB human dorsolateral prefrontal cortex. That data was generated from 10x Bayesian data, and they compare the cancer region and the spatial clustering through Loven, ST-Learn, and Bayes-Space, these three tools. And we can see that after the histology analysis, you cannot define what kind of tissue in this histology area. After the manual annotation, which was conducted by a brain expert, then they have performed that they define this area was called several domains in this brain tissue. Then, using the cancer region and the spatial clustering, we can find that the Bayes space has a greater tissue distribution, and compared to the spatial GCN, it shows the best gene expression profiling in spatial region. So they find that both GCN and Bayes space reveal a spatial domain that agreed better with the manually annotation tissue layer than Loven and ST-Learn. So they find this tool is very valuable to define the specific brain region. Then they also analyzed the Moran and Greer's C-value for the spatial variable gene, and they find that compared to the SPARC and the spatial DE, spatial GCN shows a higher level. Moreover, they want to observe the spatial gene expression pattern through the different genes. Then they find that in human brain, the domain 0, which expresses the CAMK2N1, it shows a higher level, and domain 1 shows the PCP4 gene expression, and domain 3 shows the NEFM gene expression. So using this gene expression through the different domains, they can define the specific gene expression, and it will be located in the different spatial region. So they further want to compare which spatial variable genes which was compared to now available gene expression profiling. So they compared spatial GCN to spatial DE and SPARC. And we can see that through the manual annotation of the human brain, we can see there are several domains, and it will be defined in the several layers. And compared to the spatial DE and the SPARC, the spatial GCN shows the great annotation, and it will show several different gene expressions in different areas. They want to use the meta genes, which will be detected by spatial GCN. For example, the domain 1, domain 2, and domain 4, domain 6. They will show the several gene expression profiling, and they using this different gene profiling, which called the meta gene, it will be annotated for the different gene expression, and it will define which location is specific domain. Then we can understand what domain is important for this gene expression. So they have applied this tool to the human brain and using the 10SpaGCN data. Next, they want to observe whether this tool can be observed in the mouse brain. So they also use several tools and find that the spatial GCN can define the specific mouse brain area, which will be performed better than other tools. Moreover, they also can use the subdomain and find the specific domain region of the small mouse brain. Further, they want to use the Allen Brain Institute reference atlas diagrams to understand the mouse cortex and the subdomain. Then they have found that this is the reference mouse brain cortical layers, and the right side is the spatial GCN subdomain, which is manually annotated, and find that this tool can perform very similar results to the mouse brain reference. So this tool is a very powerful tool to identify the same color tissues from the histology analysis. Then we can understand which gene may be important in this tissue area. Then they also compare the Moran and Gears C value and shows that the spatial GCN has higher value than SPARC and the spatial DE. Then this result shows that spatial GCN has a more powerful tool for the spatial gene expression. Then they also define the gene expression profiling through the different domains and find that the spatial gene profiling of SPGs, which is detected spatial GCN for domain 1 is PVALB, and for domain 5 is NRGN gene. for the MEM8 is TRIM62 genes. So we can, through this analysis, we can understand which area shows the most abundant gene rather than one bulk analytic. Then we can understand in which tumor, which brain area is important. This gene will maybe play an important role in this specific area. Then this is another example, shows that the spatial gene expression pattern of genes KL, K6, and BP, and ATP1B1. They also use the meta genes, which defined as KL, K6 positive plus and BP positive and ATP1B1 negative. Then using the meta gene analysis, we can define the brain tumor in a more specific region. So from this experiment, they have established the joint spatial domain, which detected through the multiple mouse tissue, and using the SpaGCN, then we can see the beautiful map of the mouse brain. So this tool is like, I consider this tool is like a Google map. Then we can know when I have expressed this gene and I want to know what kind of the gene expression profiling in my neighbor. So I can understand the gene expression profiling in the different region. That may lead us to more understanding what is the disease causing effect. So the summary is that they have established the SpaGCN and make its evaluation to compare with the Spatial DE and the Spark. These are available tools, and they have applied this method to the human primary pancreatic cancer cells through the spatial transcriptomics data, and this tool also used to understand the gene expression profiling in the complex human brain and mouse brain using the 10x data. So the advantage of SpaGCN is that because it's guided the differential gene expression analysis, then this can detect the gene enrichment, and in the identifier domain, it means the specific region of histology analysis. Then this enriched spatial gene expression pattern were more powerful than competing methods. And this data is transferable, and that is to utilize to study the spatial variations of gene expression in several data sets. For example, in this paper, they have used the spatial transcriptomic data set and the 10x data set. And it is computationally fast, platform independent. It will make it easier to define the diverse spatial results transcriptomics analysis. So in this paper, they has make a breakthrough is that they have understand the spatial gene expression, and they have first identified the spatial domain by integrating gene expression profiling, spatial location, and histology analysis. Then they will represent the spatial dependency of these data. So we can understand the different gene expression in different regions. And this is a new breakthrough to let us know, because one tissue has several parts of cell. One in cancer, they have some immune cells which were infiltrated into the specific tissue and some blood vessel and cancer cells, stroma cell. Through this technology, we can understand what kind of gene expression in different regions. So we can understand what is the pathological effect of different cells in different disease. Thank you for your attention. Do you have Okay, do you have any question? Okay, do you want to take a break? Okay, let's take a break for 5 minutes.